everyone. My name is Trinity Estilla, and today I will be talking about Yersinia pestis. Yersinia pestis is a bacteria that is known to be the causative agents of plagues, the most common being the bubonic plague. It is said to have emerged into the world about 5,000 years ago and was founded by French physician Alexander Yersin. Did you know over more than 200 million people died from the bubonic plague? It killed off about one-third of the European country during the mid-1300s. This is due to the fact that the bacteria is airborne, meaning that it can be transmitted through sneezing and coughing. It is also transmitted through the means of rats and domestic pets, such as dogs and cats. Their coats contain fleas with the bacteria, and once a flea bites that of a human, the bacteria is then administered into the bloodstream. This soon creates lymph nodes, which are called buboes, hence the name bubonic plague. Patients who have fell ill to the bubonic plague will have raised lymph nodes. They can be found in the neck, armpit, and groin area. The lymph nodes are red, hard, but tender to the touch. Patients will often experience high fevers, chills, muscle aches, and headaches. They may also suffer from extreme weakness, making it hard for them to do everyday activity. Luckily, antibiotics are able to tend to this. The most common ones are streptomycin and chloramphenicol. Streptomycin is known to be the most effective against Y. pestis, while chloramphenicol targets tissues infected with Y. pestis. Both are to be taken orally. With streptomycin, it is recommended for patients to take 30 milligrams for about 1 to 2 months. On the other hand, for chloramphenicol, patients are to take 50 milligrams for about 10 days. Yersinia pestis is a rod-shaped and non-motile bacteria. Because it cannot live without a host, it exhibits parasitic behaviors. And since it develops in the flea, it cannot be transferred to the host without the bite of a flea. When we look into the bacteria, we can see that it contains chromosomal DNA, the CO92 chromosome specific to Y. pestis. In addition, it is distinguishable by the PMT1, PCD1, and PPCP1 plasmids. These plasmids are what makes the bacteria special. They are the mutations from the bacteria Yersinia pseudotuberculosis. However, further research has shown that Yersinia pestis is a clone of Y. pseudotuberculosis. In order to test this theory, scientists took 76 strains of Y. pestis of all three biovars, Antiqua, Medievalis, and Orientalis, that had been isolated from humans, fleas, and small mammals in diverse countries. Gene fragments were sequenced from 36 strains, and IS-100 typing was done with the other 49. In addition, 13 strains of Y. enterocolitica of various bioserotypes and 12 strains of Y. pseudotuberculosis of serotypes 1 through 5 were also used for sequencing gene fragments. The genes were PCR amplified from chromosomal DNA of Y. pestis or Y. pseudotuberculosis by using primer combinations. The table to the left shows the 36 strains of Y. pestis that were tested as well as the strains of Y. enterocolitica and Y. pseudotuberculosis. The strains came to withhold identical alleles for all 6 gene fragments. Identical THRA1, GLNA1, and MANB1 alleles were found in Y. pestis and some strains of Y. pseudotuberculosis as well. And yet, these alleles were not the only ones that made Y. pestis similar to Y. pseudotuberculosis. Evident on the table to the right, for the 3 other gene fragments, Y. pseudotuberculosis alleles were found that encoded polypeptides identical to those from Y. pestis. This can be seen for polypeptides TRPE and DMSA. These observations tell us that Y. pestis evolved from Y. pseudotuberculosis. With this information, phylogenetic trees became easily constructible. It shows the alleles present in the 13 strains of Y. enterocollidica, identical again, and remains consistent. The tree gives us a banded patterns of IS 100 in the 49 Y. pestis strains that were sequenced. Despite the comparisons between Y. pestis and Y. pseudotuberculosis, the gap in its phylogenetic tree is large. Yet, there's only a 4 to 20% difference between the two bacteria. Y. pestis is a clone of the bacteria Y. pseudotuberculosis. Its evolution shares a strong sequence homology in which the alleles of the two are practically identical. However, when studying the bacteria, there were sequences missing and lack thereof. The lack of sequences gives the bacteria a broad range in its age of evolution, 1,500 to 20,000 years. Since its recent breakthrough as a clone, this range of evolution may be too extensive. It is unknown to how and what pushed Y. pestis into being. This is because it infects a variety of small animals and that it is not primarily a human pathogen.